Well, thank you to ACAMP for inviting me. I really appreciate this opportunity to talk to you about some of the research that I'm doing. Specifically, I wrote the longest title I could possibly think of. It makes me look smarter, I think. But in general, we're looking at methods of controlling drug delivery in a local position in the body. A lot of work has been done previously in tuning various polymer systems that are smart. They can sense temperature changes or pH changes, or they might degrade under certain physiological conditions at a certain rate, thus mediating the release of their drug or their therapeutic molecules. However, the idea I was rather looking to employ looks at using tissue-resident enzymes as the main modulator of releasing peptide therapeutics in order to address various disease states. So it's hypothesized that the self-assembly of ion complementary peptides will provide a novel platform for developing injectable or minimally invasive bioresponsive hydrogels, or hydrogels that can respond to the environment that they're in for drug delivery techniques. It's hoped that the rational design of these materials will allow us to do this, and that in order to do that we need to understand the influence of peptide composition on the assembly process of these self-assembling peptides. We have to understand the plasma compatibility of the peptides in plasma protein situations, and we also have to understand the in-gel enzymatic activity of the tissue-resident enzymes. Well, strategically speaking, peptide therapeutics have been well recognized as being highly active, highly specific, while minimizing drug-drug interactions and minimizing cytotoxic effects. However, the main hindrance to using peptide therapeutics has been the fact that these peptides are usually easily cleaved within serum by various proteases. Some of the pros and cons of using peptide therapeutics have been documented by Frost and Sullivan. You can see some of the advantages include high activity, high specificity, little unspecific binding to molecular structures other than desired targets, less accumulation in tissue, etc., often very potent, meaning you can use less of them to get a similar therapeutic response. The disadvantages: you can't take them in through the oral pathway. Injection is required. They're not very stable by themselves. They're difficult to deliver. However, even despite these disadvantages of using peptide therapeutics, it's forecasted that peptide-based pharmaceuticals are expected to double the growth rate of the overall pharmaceutical industry. Frost, Sullivan, and Palmer have reported in 2005 that currently 40 marketed peptide therapeutics were available worldwide. 270 were in clinical phase testing at that time, and they had over 400 in advanced preclinical. So you can see the pipeline for these is growing quite a bit. Moreover, it's been acknowledged that it's not just enough to find the proper peptide that does the therapeutic job, but a novel delivery technique is really required in order to maintain their activity at an appropriate concentration and in the right location. And even some companies have started to invest in peptide therapeutics. So there's sort of a market there. People are starting to move into it. There's a big pipeline of therapeutic protein or peptides out there. I'm not necessarily interested in discovering new peptides, just the delivery of them. It's been recognized already that self-assembling peptides have the potential for delivering labile proteins, peptides, or genes. However, very little work has been done on developing the peptide-based hydrogels for site-specific delivery. So we're hoping to develop injectable bioresponsive hydrogels. We're hoping to put NRC and Alberta at the forefront in this work. So the key challenges in developing these materials revolve around these points. We don't quite fully understand the limits of self-assembly for these self-assembling peptide domains. We don't quite understand yet the material-induced foreign body and inflammatory responses toward these materials. We don't understand if they'll create or initiate thrombogenesis. We don't understand quite yet the disease or injury-specific release of the molecules. And we have to understand the non-specific degradation of peptides by other proteins that we don't want, or other enzymes that we don't want to degrade the peptide-based delivery vehicle. Well, in general, it's understood that upon implantation or introduction of a foreign body into the host, there's a rapid adsorption of proteins that occurs at the interface between the tissue and the material. It's this interfacial adsorption of proteins that then allows for the interaction or facilitates the interaction between the cells within the body and the material. And depending upon that interaction, it can either lead to an inflammatory response or whatever. In this case, it goes to the end result, which forms the acellular collagenous bag around the foreign material within the body. And you can imagine if you're trying to deliver a drug from this material, as soon as you form this acollagenous bag around it, you're going to inhibit the release rates. Another idea here, another point to consider is that depending upon the molecular level cues, whether it's C3 activation through the interaction of surface-bound hydroxyl groups, or if it's the intrinsic activation in the coagulation cascade versus the surface contact protein system, either way we can lead to inflammatory response, or we can lead to clot formation, which are both deleterious host responses. So these molecular level events need to be understood with respects to the peptide composition. In general, we were looking at RADA4. These are amino acid sequences. They've been shown to form nanofibers that are quite soft. At high concentrations, they form a gel, and they can form gels that contain up to about 99.5% water. The enzyme of interest that we're targeting is matrix metalloprotease 2. The main reason is that it's naturally present during many physiological events, including wound healing. It's also present during cancer or high concentrations within tumors. It's highly regulated. It's located in the cellular periphery, which is where we would inject our peptide scaffolds. It's extremely active, and it's also associated with various types of cancer. Furthermore, we have an understanding about the spatiotemporal profile of MMP2, and we can hopefully utilize that to control the release rate of any tethered peptide therapeutics to our RADA. So the basic domain looks like this: RADA4, which self-assembles into nanofibers, an MMP2 cleavable domain, and then a peptide drug. This paper in specific used RADA4 on the other side to look at how you can degrade the hydrogel itself by cleaving the MMP2 domain.